Hello students, welcome to your school YouTube channel. Today's topic is explain the process of DNA fingerprinting. So without wasting time, let's quickly start the video and learn. DNA fingerprinting, also known as DNA profiling or genetic fingerprinting, is a technique used to identify individuals based on their unique DNA characteristics. Here's a step-by-step explanation of the process. Step 1. DNA extraction. The first step is to extract DNA from cells in a biological sample, such as blood, saliva, skin, or hair. This DNA is then purified and isolated from other cellular components. Step 2. PCR polymerase chain reaction. The extracted DNA is then amplified using PCR, a technique that generates millions of copies of specific DNA sequences. This step is crucial for analyzing small DNA samples. Step 3. Restriction enzyme digestion. The amplified DNA is then treated with restriction enzymes, which cut the DNA at specific recognition sites. This creates a unique set of DNA fragments. Step 4. Gel electrophoresis. The DNA fragments are then separated based on their size using gel electrophoresis. This process involves applying an electric field to the DNA fragments, causing them to migrate through a gel matrix. Step 5. Southern blotting. The separated DNA fragments are then transferred to a membrane using southern blotting. This step helps to preserve the DNA fragments and prepare them for hybridization. Step 6. Hybridization. The membrane is then treated with probes that bind to specific DNA sequences. These probes are labeled with radioactive or fluorescent markers, allowing researchers to detect specific DNA patterns. Step 7. Autotrophicity or fluorescence detection. The membrane is then exposed to X-ray film or fluorescence detector, revealing the pattern of DNA fragments. This pattern is unique to each individual and is known as a DNA fingerprint. Step 8. Analysis. The final step is to analyze the DNA fingerprint. Researchers compare the DNA pattern to known DNA samples or profiles to identify matches or exclusions. Applications of DNA fingerprinting. 1. Forensic science. DNA fingerprinting is widely used in crime scene investigation to identify suspects, victims, and human remains. 2. Paternity testing. DNA fingerprinting is used to determine paternity and resolve family disputes. 3. Genetic research. DNA fingerprinting is used to study genetic variation, population genetics, and evolutionary biology. 4. Medical diagnosis. DNA fingerprinting can be used to diagnose genetic disorders and identify genetic mutations. Benefits of DNA fingerprinting. 1. High accuracy. DNA fingerprinting is a highly accurate technique for identifying individuals. 2. Uniqueness. DNA fingerprints are unique to each individual, making it a powerful tool for identification. 3. Sensitivity. DNA fingerprinting can analyze small DNA samples, making it useful for forensic applications. Limitations of DNA fingerprinting. 1. Sample quality. DNA fingerprinting requires high-quality DNA samples, which can be a challenge in some cases. 2. Contamination. DNA samples can be contaminated with DNA from other sources, which can affect results. 3. Interpretation. DNA fingerprinting requires specialized expertise to interpret results accurately. Overall, DNA fingerprinting is a powerful technique that has revolutionized the field of genetics and forensic science. Its applications continue to grow, and it remains a crucial tool for identifying individuals and solving crimes. Let me know in the comments how you liked the video and what topics you would like to see in the next video.